Hello, and welcome to chapter 21, which is the immune system. And we are in part 3, where we are going to look at some of the cells in the innate branch of the immune system. We will look at the cells in this lecture, and then we'll move on in the next couple lectures to see the chemicals and antimicrobial proteins and how they work. So if you remember, the immune system has two branches based on the way that they respond. We have the innate branch, which we call non-specific, which means that it responds the exact same way no matter what invader or pathogen it encounters. As opposed to that, we have the adaptive branch, which is specific. So the adaptive branch are going to be your like your Navy SEALs. They're specially trained for one mission. So we'll find cells in the adaptive branch, but they're trained for one specific thing. The innate cells can respond to many different kinds. The innate has no memory, which means it's not going to remember if it's seen this pathogen before. It's going to respond the same way. The adaptive branch does have memory. It takes a little time for that to develop, but it does have memory so that when it responds the second time, it will respond quicker. The innate includes the first and second line of defense. So just think about this. The first line of defense are the external barriers. That's the wrapping that protects you. So that's your skin. And then on any openings to the skin, you have mucous membranes that line those openings. So the first line of defense is going to be the outermost part of your body, the skin and the mucous membranes. If an invader breaches your skin or mucous membranes, we make it in to the skin or mucous membranes, then we're going to have the second line of defense, which is internal, right? Because that's external, because it's your skin. And this is going to include some cells and chemicals that produce some processes. Some cells will kill directly, some will produce chemicals that trigger a cascade of reactions to attack this. Additionally, some cells of the innate branch can activate the adaptive branch, which kind of just means like, hey, we're a little bit overwhelmed, let me tag you in. So they will wake up the adaptive branch, and then the adaptive branch will develop a response specific to that invader. And that can include antibodies or some ways that those cells can directly kill other cells. All right, so let's look at the cells we'll talk about in this lecture. We're going to look at phagocytes. We've talked about phagocytes before. These are white blood cells that kill by engulfing the pathogen and digesting it. That I've referred to that before as the Cookie Monster method. So they gobble down all these germs. And we're going to also learn about NK cells that kill in a different way. In the following lectures, we will learn about inflammatory chemicals, antimicrobial proteins, and fever. Okay, also we have some cells, so kind of some cells that have one foot in the innate branch and one in the adaptive branch. And these are called antigen presenting cells. So these are cells, like we just talked about the macrophage, that can then take the identifying parts of the germ that has invaded and show it to the adaptive branch, and we call that priming them or activating them so they can wake up and start and train their army. So those are macrophages and dendritic cells. All right, so let's look at phagocytes first. So we talked about that. Phagocytes kill by engulfing the bacterial, drawing it in, and then having their digestive enzymes attack it and destroy it. Most phagocytic activity takes place outside of the blood. So we don't see a whole lot of this going on inside the blood. And think about that. That's because by the time an invading germ has entered the blood, now it's in circulation, it's going to every organ and tissue in your body. So what we want to do with the immune system is destroy and take down these invaders locally. So if you cut your foot and it gets infected, we want the infection to stay in your foot, not get into your blood and travel all through your body. Phagocytes, again, are non-specific. They can attack bacteria, viruses, fungus, whatever comes in and doesn't look like it belongs there. Phagocytes also can finish destroying dying cells that belong, they're cells of your body, but they are on their way to death, and phagocytes can take care of that. So we're going to look at two phagocytes, neutrophils and macrophages. We've talked about these before in the blood system and in the lymphatic system. So we'll talk about them again. And I just want to remind you that neutrophils kill by phagocytosis. They also can kill by secreting defensins. Defensins are going to be one of those chemicals, the antimicrobial chemical that we talk about in the next lecture. So you're going to see throughout this immune system, we're taking what we learned from one lecture and applying it in the next. Macrophages are going to kill by phagocytosis as well, but they are what we call antigen presenting cells as well. They will take a chunk of what they have killed and go over and show that to cells in the adaptive branch and wake them up and tell them what to train for. Now, here's neutrophils. We've seen them before. These are the big white blood cells here that have the multi-lobed nuclei. So there are 4 neutrophils in this blood smear. They're abundant. They make up the majority of circulating white blood cells. They are your first responders. So these are your boots on the ground soldiers. They are delivered in mass, and within 12 to 24 hours after bacteria. Remember, neutrophils love bacteria, and they're best at fighting bacterial infections. So within 12 hours, they're on the scene and begin to phagocytize. Now, they're like that Cookie Monster too. So they're just eating up everything, making a giant mess, really. But they are quick and can phagocytize bacteria, or like I said, cellular debris, so old cells, dying cells, things like that. And then the neutrophils usually die after phagocytosis. So they're going to leave a giant mess, but they're going to have killed some things. The macrophages will come in later after the neutrophils and clean up the mess that the neutrophils left. They will phagocytize the dead neutrophils as well as any other invaders. Neutrophils can kill in the blood cell, but remember, they have this superpower called diapedesis, which means they can leave the blood and enter the tissue and then kill in the tissue. They also secrete defensins. Defensins are antimicrobial proteins that disrupt the bacterial cell membrane and kill the cell. So I put this picture of, I don't know if you all are Katniss fans from the Hunger Games, but imagine defensins are kind of like a poison arrow. So neutrophils can also kind of shoot these over at the bacteria, and like an arrow will poke holes in the cell membrane, and then the cell membranes will dissolve, and then the cell will die. All right, here's a macrophage. So this is a monocyte after it leaves the blood and enters the tissue. It is going to use these long tentacle arms to grab the bacteria and reel it in. Macrophages can phagocytize bacteria, cancer cells, dead neutrophils, cellular debris, viruses, anything that doesn't have a cell marker. They're going to come in after the neutrophils and then kind of clean up the mess. They're also going to take some of these, the antigens, the disease-causing parts in the pathogen, and show it to the T cells and B cells. Those are T lymphocytes and B lymphocytes in the adaptive branch. This is just kind of showing you what antigen presenting looks like. So here's your macrophage. It it's digesting the pathogen, or these little square things. It's digesting it. And then it's going to come over here and kind of say, hey, T cells and B cells, let me show you what I found. And then the T cells and B cells will recognize it. They have, see, this is like a little receptor that fits the square. And then they will kill in two different ways. So we'll see B cells produce antibodies that will kill or tag a cell to die. And cytotoxic T cells do exactly what their name says, cytocell toxic death. So they can directly kill an infected cell. We've talked about eosinophils and basophils before. These are other white blood cells. They also can perform phagocytosis. It's not their main job. Remember, eosinophils, they mostly function in parasitic worm infections. Basophils, we have talked about basophils being active in allergies. We're also going to see that basophils and their cousins, the mast cells, are active in producing inflammatory chemicals to help the inflammation process. This is a dendritic cell. We talked about these in the lymphatic system. These are also antigen presenting cells, as you might imagine. They have all these big long arms. They can grab a piece. Now, they're not phagocytes. You can see they are antigen presenting cells. So they can grab a hunk of whatever has come in that's not supposed to be there, and then take it over to the B cells and the T cells in the adaptive branch, show it to them just like the macrophages did, and help activate them and begin a more intense immune response. Phagocytes have to recognize a pathogen, and so don't get too lost in this microbiology here, but just know that the outsides of pathogens are often covered in a special kind of carbohydrate. And so the phagocytes can recognize that carbohydrate and say, hey, this is not supposed to be here. But some bacteria are pretty smart, and they have another coating outside, kind of think of like they were in a costume, covering up their signature, their carbohydrate. And so then they kind of sneak around because they're wearing a costume that makes them kind of blend in with everybody else. And then they can evade detection and proliferate and make us sick. So our immune system can coat these pathogens with something called opsonins. And that is provided by something called complement proteins, which we'll get to, and antibodies. So essentially, these two substances, the complement proteins and the antibodies, are going to coat them with this thing called opsonin that makes them easier to identify. They can also tag or coat dying cells which need recycling, right? So if a cell that has been infected, we can tag you, or pathogens that try to form capsules. So essentially, we're just like, imagine you have a bacteria, and it looks like a bacteria, has its signature carbohydrate, but then the bacteria wears a costume, so it blends in. But then the complement or the antibodies can still recognize it, and then they tag it. They don't kill directly, but they tag it like put a dunce cap on it, put a big red target on its back and say, hey, we found this hiding in the bushes. And then the phagocytes can then say, oh, now I see it. I will come and attack it and gobble it up. All right, now let's look at these NK cells, natural killer cells. That's a great name for a cell, don't you think? These are cousins of the lymphocyte. So they are a type of lymphocyte, and they are like the police. They are constantly circulating throughout the lymphatic system and the blood, searching for things that aren't supposed to be there. They kill by releasing an antimicrobial protein called a perforin. So think about that. Perforin, it sounds like it makes pores. And what it does is punches a bunch of holes in the cell membrane, and that triggers the cell to die. If you've ever seen a like a marinating knife that has all these little punch holes in it that makes your meat more tender, that's what these perforins can do. So they're going to poke a bunch of holes in the cell membrane of the cells that aren't supposed to be there and induce apoptosis. So apoptosis is programmed cell death, but we're just going to kind of hurry it along. NK cells do not kill by phagocytosis. Right? They only kill by releasing these proteins that punch the holes all in the cell membrane and then induce the cell to die. Interestingly enough, NK cells can kill cancer cells, virus cells, and other non-self cells. So they're very important. They also secrete some chemicals to enhance the inflammatory response, which we'll talk about coming up. When NK cells are impaired, and they don't mutate it or they don't do their job, we can develop autoimmune disorders. And autoimmune disorders are disorders where the body doesn't recognize itself and starts attacking the self, good cells, and killing them instead of a real pathogen. These are what NK cells look like. So they look kind of like, you see, like they have sometimes a circular nucleus like a lymphocyte, but sometimes a kidney-shaped nucleus like a monocyte, but they do have these granules. Remember, both monocytes and lymphocytes are agranulocytes. They don't have the granules in their cytoplasm, but these do, and these are the granules in here that contain that perforin that they throw out and punch a bunch of holes in the cell membrane of the invading cell. All right, and then we talked a little bit about antigen presenting, so I just want to show you here a monocyte, which is a white blood cell, an agranulocyte in the white blood cell, can differentiate into macrophages or dendritic cells. Both macrophages and dendritic cells are antigen presenting cells. So they both end up with those long tendril arms like an octopus that can grab parts of the bad guys and pathogens and pull them in. Macrophages enter the tissue and they kill by phagocytosis. Like I said, they can take a chunk of what they've digested and show it to the adaptive branch. Dendritic cells do not phagocytize, and they only live in the epidermis. That's the outer layer of your skin. So the dendritic cells only live in the skin, and they are there monitoring for breaches into the skin, and then they can grab a hunk of it and present it over to the B and T lymphocytes of the adaptive branch of the immune system and activate that branch. This is just showing you that we have neutrophils that can attack bacteria. We have dendritic cells that can grab a chunk of it and present. And then depending on what it is, we're also going to see mast cells working, NK cells that kill by apoptosis, and we know similar eosinophils are kind of like neutrophils, but they work mostly on parasitic worms.